So this afternoon I'm going to tell you about our progress with PURA Syndrome Biobank and a little bit of information about some of the issues in collecting clinical samples for research. So first let's start with what is a biobank. A biobank is actually a physical repository of samples that are collected for research purposes. And the idea here is that investigators who are studying PURA syndrome would be able to request samples from the bank to carry out their research studies. So the bank would follow all of the approved ethical protocols, making sure that everything's being done properly. And this includes informed consent, which should be obtained from the patient, or in the case of minors, their guardian. And it's very important that all of the appropriate regulatory guidelines are followed. So this is actually like a bank where you're making deposits. You think about it like a safe deposit box. And then over here, we have an example of cells being collected here in the freezer. And these cells, you can see, could be multiple samples from a particular patient or several patients. So this is a working diagram of what the biobank looks like in our initial stage. In Southampton, we have the patient registry located, and then they will also have a site for banking of samples. And this will be a site where samples from across the UK can be collected. We will also have two additional sites. One will be at Temple University in Philadelphia, and this will be the storage site for collecting samples from across the US. And then we will have a site at the Helmholtz where they will collect from the EU countries. And then as we expand and scale up, we will determine how we can handle samples collected from the rest of the world. So this is a diagram showing the workflow of the biobank. So the first step is the informed consent, obtaining of the signed protocols. What you don't see here is the whole approval process for the protocols, making sure that we have the ethics in place. But usually the patient contact begins with the clinician discussing with the patient the project, the purpose of the study. This could be the clinician themselves, or it could be a biobank coordinator or research coordinator. The next step is the collection of the samples. This could be something simple like obtaining blood or urine, or it might even include biopsy material or something that is obtained during surgical procedure. Next, we have processing of the sample. This could be preparation of the material, aliquoting of the material, and then appropriate storage of the material. And this depends on the type of tissue, what the storage requirements will be. Then individuals will request samples from the bank for their study. And then the bank will be able to distribute these materials to the investigators. And then the investigators will analyze the material in the laboratory. They will have results from their analysis, and those results will be disseminated. And they may also create derivatives, say culturing of new cell lines or new material, and that may be deposited right back into the biobank. So how can the samples in the biobank be used? Well, obviously we can analyze samples directly from the patients, but we also need to have control samples for comparison. In some cases, this might be from parents, from siblings, or from other patients, so we can compare from patient to patient. We also have a need for control samples, and these would be what we might call normal, healthy controls, though the question is what we would consider normal. We may look at samples from a single time point, or we may do a comparison of samples before the development of the disorder or development of particular symptoms of the disorder. And then one of the powerful approaches is to do a natural history study. And this is a longitudinal collection of samples over time. So you can take a snapshot of the patient's, say, serum in different months after diagnosis or every year, and compare longitudinally what differences may happen during the course of progression. And then, of course, if we have interventions, some sort of treatment for a symptom or for the disorder, then we can compare samples collected before and after treatment to see what kind of changes are taking place. So how will investigators be able to get samples from the bank? The first step is that they will need to submit a request. So the investigators will submit a request to the bank, and in this request they need to explain to us the purpose of the study and the goals of the study, how many samples they need and how much of the material they need. And then the Biobank committee will evaluate all of the requests, and we will decide what the priority is for those studies. And this will be based on the goals of the Biobank, which are to support the goals of the Foundation for Research. The Foundation will require that the results of the study of any of the samples from the bank need to be reported back to the Foundation. And we would like to see that investigators using those samples have a plan in place for future development. And what we mean by this is that the Foundation will provide these samples to help investigators develop preliminary data and get pilot study results that will help them go on to secure further funding. So in many cases, this means government funding or institutional funds that can be really large dollars in terms of research grants. But often the first step is doing smaller pilot studies and being able to collect material and perform analysis from patient samples directly. And the Foundation also will require that the results be published in a timely manner, and we would want this to be published in open access journal or freely available journal online. So what are the types of samples? There are many, many types of samples that we could collect in the biobank. And one of our goals in our discussion here at the conference is what priority we should place on samples. Sometimes this is based on the availability and the ease of collecting the samples. Less invasive samples or non-invasive samples are obviously easier to collect and are less risk to the patient. And that would include something like blood, which is minimally invasive, and we can purify from the blood different components, including serum or plasma or PBMCs. Different types of white blood cells can be isolated from the blood. Urine, which is a very rich source of metabolites. We can also collect biopsy samples. This could be skin biopsies, which could then be used to grow the pluripotent stem cells that we heard about a little bit earlier, or other biopsy materials. We can culture cells and freeze down these cells so they're available for further use. CSF is a very valuable source. We would mostly acquire CSF if it is a collection that's being done for a diagnostic purpose, and then the extra CSF that would normally be discarded, we may be able to collect from the clinical lab and deposit into the bank. And then, of course, we can also collect autopsy material, which is a very valuable source for us to see what's going on, especially in the brain. There are many, many other types of tissue, and we will need to prioritize which ones to put into the bank first, and this list may expand over time. So the different tissues that we collect have different storage conditions. Some are very stable and can be held at room temperature indefinitely. Others need to be kept frozen or in liquid nitrogen. So each of the different types of material has its own requirements. So before we all line up and start donating our material, it's important to think a little bit about the ethics involved and what sort of risk and reward there are for participating in clinical research. So one of the first questions that we ask if something is going to be done to a patient is whether or not the ends justify the means. Is there a procedure or is there a treatment that we would try on a patient, let's say treating one patient that might help hundreds or thousands of patients? Is it ethical to do that? So there are some universal principles for human subjects research, and these have come over many years, I would say over the last 50 years or so. There's been a consensus that's been reached in most developed countries, and that are abided by all of the academic institutions that are participating in the banks. And these key principles include the benefit must outweigh the potential risk or harm to the patient, and this is really critical. It is not enough for society to benefit. It must actually benefit the specific patient, the subject itself. And we need to make sure there is no coercion of patients to participate. They should freely decide to participate themselves. And another principle that's very important, there's something known as standard of care. This would be the treatment that we know works for a particular disease. Let's say you get an infection and we have an antibiotic to treat that infection. Then it is an obligation for a clinician to treat a patient with treatment that is known to work for that disease. And it is not ethical to withhold that treatment for the sake of a study. So the patient's safety and the patient's health is paramount. So the answer to this question, do the ends justify the means? In the case of human subjects research, the answer is no. So I want to talk a little bit about sources of conflict of interest. Some of these may be fairly obvious. We think about financial conflicts of interest, and most of us immediately know what we're talking about. But there may be other more subtle types of conflicts, like professional conflicts, conflicts that you have in the relationship with your clinician. Personal conflicts, this can be somewhat more subtle, and we may not even be aware of our own conflicts. But all of these can affect our behavior and our decisions to participate in research. So let's first, sorry to any clinicians, but they're an easy target for potential conflicts. So we know that clinicians may receive salary, bonuses, travel expenses, honoraria, things like that, different speaking fees and whatnot. And they can have research funded by a particular sponsor. So these are conflicts of interest, financial in nature, and they're easy to track and easy to disclose. And in the interest of transparency, there's been a big push in the United States to make sure that these disclosures are available for individuals to see. We're getting a lot better at it, but I'm not sure that all of the potential financial conflicts are disclosed in a timely manner. Another example of this is a clinician who might own stocks or shares in a particular company, or they may be on the board of directors or scientific advisory board for a company, and you can see how they may have a vested interest in the outcome of a study of a particular drug or something associated with a sponsor's work. We should also remember that the sponsors, and the classic example is the big pharma, they report to shareholders. Big pharma does not report to patients. They don't report to doctors. They're for-profit companies. The way they're structured, they need to make money in order to survive. This is not a bad thing because we do need the funding for these studies, and Big Pharma often has deep pockets to fund the sort of clinical trials that no one else is able to fund. But they are incentivized by profit, and this can have unintended biases. Now here's a personal conflict of interest, and it really depends on your perspective and where you're sitting on what the potential conflict might be. But in the case of most of us in this room, we have a very strong desire to find a treatment for PURA syndrome, and this may color our perspective and how we see whether a drug may work, how much hope we put in a new treatment for our child with PURA syndrome. And this can change your perspective and change your bias toward the drug. In another example, we know that there are many complementary and alternative medicines that are proposed to work for all kinds of things. All you have to do is get online and you can find a million cures for everything. Do they really work? So on the one hand, you may have a strong personal bias to try to help your child, or the people that are selling these different treatments have a strong financial bias for you to buy them. The other side of the coin is that you can see that people may be taking advantage of desperate patients and families looking for any type of cure at any cost. So we also need to keep in mind that societies and foundations, patient organizations, are not immune from conflicts. This is an article published in the New England Journal last year, and they examined patient advocacy organizations. Most of these are fairly large organizations, and they found that 83% of these organizations reported receiving financial support from industry. This might be support for conferences. For example, the conference that we're at here for the PURA Foundation. As the foundation grows and the conferences expand, we may see that the foundation receives support from pharmaceutical companies or other drug manufacturers, biopharma, in order to support the activities at the conference. And you can imagine that there might be some attempt to influence. For example, if a major sponsor wanted to donate funds to support the conference and then requested the conference organizers allow someone to come and speak from that company, would the foundation say no, or would they allow them to present? And these are questions the foundation should be discussing at the board level and with their conference committee, and those are decisions the foundation will need to make. So there are many forms of unconscious bias, types of bias that we may not be aware of, but nevertheless they're there. They may be unintentional, but they can be affecting our response. And there's a lot of research going on with these different types of biases that I've listed over here on the right. And this happens often between the patient and the clinician for diagnosis, treatment and drug choice, and some other ways that may happen in the diagnostic process. So the bottom line here is that none of us are immune from these types of unconscious bias, and we should keep this in our minds when we're looking at how we're making decisions and how our clinician is making decisions about our care. So we really should keep in the front of our mind that there could be undisclosed influences or unconscious bias at any level and at any time. So these are good questions to ask yourself. So who is looking out for the patient? When someone is asking a patient to enroll in a study or decisions are being made about clinical management, who's speaking for the patient? And is your clinician looking out for you? We all hope that they are, and building the trust with a clinician is very important. So when a clinician or a research group is approaching you to ask you for your participation or your child's participation in clinical research, there are a number of questions that you should ask so that you understand what the purpose is. What is the research? What are the questions that they're trying to answer? What do they need you to do? What type of samples are they asking about? What would happen if you participate in a study and something goes wrong? This should all be described in the consent form. So if you read the information that's provided in the form, many of these questions are required that the answers are in the informed consent. So these are reviewed by the ethics committee. You should have an understanding of what the potential risks are. And as I said, the benefits for your child to participate in the research is more important and should be higher than the risks that are associated with participation. So with children, this is especially important, that the risks are minimized and the benefits are maximized. You should also have an understanding of what kind of privacy, what kind of confidentiality you can count on for the material, the data that's collected in this study. This is something that's talked a lot about these days. I'm not going to go into more details. So the basic outline for the protections that any of the ethics protocols involved at the universities where we all work should include are that there is an approved ethics committee protocol, that it includes informed consent, and the informed consent, these forms contain all of that information that we were talking about. Patients should have the right to withdraw from the study at any time without any negative consequences, and also should have the right to remove their data from the study or their samples from the study at a future time. It must be in the patient's best interest to participate, meaning that there's a benefit to the patient that outweighs the risk. And again, as I said, this is especially important in children. So there are many different types of research studies. Clinical trials is a kind of classic example we all think about, where it's an intervention, testing a new treatment or a new medication. But there are many other types. For example, questionnaires and surveys. This is part of the patient registry that's being developed that you'll hear more about, where we can get more information about the clinical symptoms and different aspects of management, medications that have been tried. This will all be through questions and surveys attached to the patient registry. Biobanks, collecting the physical samples that we're talking about today. One type of study are retrospective studies. This is where samples that have already been collected that would normally be discarded, they may have gone to a clinical lab and the clinical lab is finished with their part. We can collect this existing information and put it into a resource or a bank. Observational studies, where you're following the natural history, studying how the disorder may develop and how it progresses, but you're not actually doing any intervention. These can be very valuable in understanding how the disease may manifest itself. And this is another very important area that's getting more and more attention, patient-centered outcomes research. So many of the questions that clinicians or researchers like myself want to ask about PURA syndrome may not be the most important questions for the patients and their families. Many times, patients and families are more interested in quality of life issues. What are the things affecting the quality of life day to day for the patient? These can often be the biggest challenges for patients, but they may not be the same level of excitement for a research question. Nevertheless, they're probably more important than answering some of the research questions that we may have to the families. So it's important that the foundation can set priorities for what is important to the patients. What are the key questions that are prioritized by the group? And I think that this is going to be discussed at the conference later on. So conflicts of interest are not a bad thing. They exist. We need to identify them. And we need to make sure that we're aware, and we need to have as much transparency as possible. Another thing is to make sure that we include perceived conflicts of interest. So for example, if I have funding from a particular company to study something, that doesn't mean that I automatically have a conflict of interest, but it might give the perception that I do. So I should disclose that relationship so that it's transparent. And then you should be able to decide whether you think that's a potential conflict, but at least you should have the information so you can make your own judgment about it. So disclosure is important, and then the goal for conflicts of interest is to put a plan in place for managing the conflicts, and this would be a way of separating some of the decision-making activities, or there are many different ways that you can manage the conflicts, but the first step is to make sure they're all disclosed. So how do we identify conflicts of interest? Read the fine print and follow the money. There are many ways you can find this information. In the consent forms that you get for studies, the funder or the sponsor's name should be associated with them. So if you read those forms, you should find that information. If you look in the publications, PubMed, if you read the fine print in the publications, somewhere after the affiliations or in the acknowledgments or buried in the bottom or in the supplemental online, you will find disclosures of conflicts of interest. Some journals, like the New England Journal of Medicine, for example, has a form that has to be filled out by each investigator. that list all the different types of conflicts, and these are all submitted as attachments in the supplemental to the article that you find online. And each of the clinicians, each of the authors will fill out their own conflict of interest form. In presentations, you should see a disclosure slide for the presenter to tell you about any of their conflicts. I have a disclosure slide at the end of my presentation. Meeting brochures and pamphlets. Any of the sponsors or the funders of the conference, any of the donors, should be disclosed in the material that you're seeing distributed here at the meeting, so that you know where the funding came from to support our activities here. Educational materials. If a company is sponsoring educational materials, that should be in the brochure, in the materials directly, so that you can identify that that sponsor funded the material, and then you can ask the question whether or not that sponsor may have influenced the content of that material. You can go to the clinicaltrials.gov website, where they have a list of all of the clinical trials in a database. This is a worldwide list, predominantly used by the US, though many other countries use it as well. And they will have sponsors lists in there as well, so you can see any clinical trial that's ongoing or completed, who the sponsors of the studies were. And as I mentioned before, there are now databases online which are reporting any of clinicians' financial information related to studies. So I didn't want to finish my presentation without talking a little bit about research, because that's what I do most of the time. Though the foundation asked me to talk to the Biobank, I decided to include this slide here about the PURA mice. So we have a line of transgenic mice that have one normal copy of the PURA gene that produces PUR-alpha, and one mutated copy of the PURA gene, which creates a short, truncated mutation in the protein. So we can compare what the brains of the wild-type mice, the normal mice, look like, which are over here on the left, compared to what the brains look like in the PURA mice. So these are sections of the brain. So what we do is we take the brains from the animals and we fix them, and then embed them into paraffin and make really, really fine slices of them, so we can look at the slice of the brain under the microscope. And we can use immunohistochemistry to stain for the PURA gene product PUR-alpha. So the brown staining that we're looking at here, and you can see these individual little cells. In the middle of the cell is the nucleus, as we heard Dirk talking about. And then in the cytoplasm, you can see this brown staining around it, and that's the PUR-alpha in the cytoplasm. And these are all neurons, and this is a region of the brain called the hippocampus, which is deep in the middle of the brain, and it's involved in learning and memory. So you can see these really, really plump cells, very healthy-looking neurons with lots of PUR-alpha in the cytoplasm. And then down here in the lower panels, we can see a region of the cerebellum. This is the back of the brain, and it's involved in controlling movement and motor coordination, walking. And there's a layer of cells in here we call the Purkinje cells. And you can see them at higher magnification here. And these little cells, again, the nucleus, which is clear or light blue, and then the PUR-alpha you can see in the cytoplasm of those cells. And then I think you can see these little processes coming out like a tree, the branches of the tree, and these are the dendrites of the neurons. And again, you can see the PUR-alpha staining there. If we look in the heterozygous animals, the mice that have PURA syndrome, we can see that some of the neurons here are lacking the staining for PUR-alpha, and then the ones that do have PUR-alpha, they have less PUR-alpha than we see in the wild type. So there's a reduction in the amount, and then some of the cells, the absence. And you can also see there are fewer cells in this region if we count the density of the cells here compared to the density of the cells here. And the cells in the PURA mice are not as healthy, not as plump or as full as they are in the normal mice. Here's an example in the Purkinje cells, and you can see this nice staining that I showed you, like the tree branch. And then over here in the PURA mice, you can see that a little bit of staining, but then some cells that have no staining at all. So why am I showing you this? Well, if we think about how we can learn more about the function of PUR-alpha in vivo, we can use the mouse as a model and compare the wild type to the PURA mouse to do some preliminary experiments. So we can create, if you will, a kind of biobank of the samples from the PURA mouse. And in fact, we had a really productive discussion yesterday, and we're going to continue that and do some goal setting tomorrow morning about what we want to accomplish in the next year, till next year's meeting. So we have two ongoing projects I'll mention. One is an RNA-seq project with Drs. Borelli and Hunt. And in this case, what we're doing is taking a slice of the brain, and this slice of the brain we're going to use to process for RNA sequences. And this is basically the expression of all the genes in the cell. And we're going to take a slice of the brain from wild-type mice and compare it with a slice of the brain in the exact same region as the PURA mice, and see what kind of changes we see between those two samples. And we're going to take another slice from the other half of the brain, so they'll be identical slices, and we're going to study the metabolites. Metabolomics is a very powerful approach. Those of you who were at the meeting in Philadelphia, you may have heard Jerzy Adamski speak. Adamski and Niesing Labs, they will both be working on this part of the project, and we're going to identify the metabolites. So we're going to have the gene expression and the metabolite expression in parallel from these two pieces of the brain. The other thing that we can do in the mice is we can collect blood and urine. Don't ask me how to collect urine from mice. It's not so easy. You can't just give them a little cup and ask them to go into the corner and provide you with a sample. They just won't do it. Nevertheless, we do collect this material, and this means that we can compare our results from the metabolomics and the RNA-seq with the metabolic profile from the blood and the urine. So what do we do with this information? We are going to try to identify potential biomarkers from blood and urine, which are less invasive and we can obtain from our patient biobank, that can be markers for disease progression, diagnosis, or other purposes. So the mouse model is a very powerful tool for us to be able to identify pathways that may be altered in the PURA patients. So back to the biobank and what our next steps are going to be. First and foremost, we recognize that we must protect the interests of the foundation. This means the interests of the parents and the patients. The foundation feels very strongly, as they should, that the foundation must retain control and ownership of the biobank material. We need to find out the best structure that is compatible across these different countries, and whether or not we need separate protocols or separate administrative structure in certain countries, or whether these can be universal. We will have a biobank committee. The biobank committee will be including clinicians and researchers, a representative from the patient registry, because we will be tying the biobank with the registry, board member, parent representatives, and we want to make sure that there is international representation to reflect the foundation. So where are we now? Well, we have identified sites, as I mentioned, in Southampton, Munich, and Philly that will be housing the samples for the biobank. And in some cases, this is the easiest thing to solve. Some of the other parts, the regulatory parts and the protocol approvals, and integrating with the patient data registry are much more complicated. Our first priority will be the banking of retrospective existing samples to make sure we can obtain them while they're still available from different diagnostic labs. And then our immediate goal is to then obtain the regulatory approval that we need to begin the retrospective sample collection. We will be having planning meetings so that we can set the goals and the priorities for the next few years that are coming. And we will set these goals and priorities based on supporting the immediate research priorities. So one of our goals at the conference here when we meet is for the research group to determine what our goals are for the coming year, and then we need to make sure that the biobank will be able to support those goals. We need to work on integration of the biobank with the patient registry, and we'll be working on that through the rest of the meeting as well. And then we need to look ahead to where we want to be in, say, 3 to 5 years. And this most likely involves a natural history and longitudinal banking of patient samples. So I would encourage you all of the PURA parents and those of you involved in the foundation to get involved. Pick some areas that are appealing to you, that may suit your skills or that you have an interest in, and do whatever you can in volunteering your time. You're here at the meeting, which is wonderful. You can hear more about fundraising and some other things that are important to do, and do whatever you can to raise awareness and educate others about PURA syndrome. So I want to put a call out for parents who want to be more involved. Consider volunteering your time to participate in the PURA Syndrome Biobank. We are looking for parents that can help us serve as liaison for one of the biobank sites. This would mean serving as a point of contact for parents, clinicians, and clinical labs. There's a lot of behind-the-scenes work in identifying where the samples are, obtaining the informed consent forms, getting them signed, getting them sent back to the central location, filling out the paperwork at individual labs, making those contacts. We need to get the samples brought in from different sites. There's a lot of shipping and other logistics. And then here's my disclosure slide. So my research in the lab, which I didn't really talk about today, is funded by the NIH. And I also have a contract with a company, Excision Biotherapeutics, for preclinical studies on HIV CRISPR. I serve as an advisor without any financial compensation for Excision and for the PURA Syndrome Foundation. And I don't have any other financial relationships to disclose. I know this is kind of a wordy slide, but there's a requirement that we provide detailed information from my medical school, and this is approved text for that. And then I actually am a patient with a rare disease myself. I have a form of vasculitis known as Churg-Strauss syndrome, or EGPA. And I am a patient advocate without any financial compensation for a research network that's patient organized for the Vasculitis Foundation. Rare diseases are very important, and we need to make sure we work together to raise awareness and bring resources for research and treatment in all rare diseases. So then I just want to show you, here's a slide of the lab in Philadelphia where the meeting was held last year, and we don't allow any children in the lab, nobody under the age of 18, so these little guys must have snuck in the middle of the night to take this picture. Here are a number of individuals at Temple that are working on the PURA project. We've been studying per alpha for over 25 years in the laboratory. And I'll just mention Brittany is a graduate student in my lab. She's here at the meeting, so please take the time to talk to her. And then finally, I just want to reiterate the support that we've had from the NIH over the years, many years, and very generous support. Much of the research that you're seeing is funded by universities and governments, and this is a great opportunity for the foundation to be able to help us do the pilot studies that we need in order to achieve more funding so we can continue our work. So I'll stop there and take any questions. Thank you.